I'm Angela Bachi, and I'm the head of the Functional Proteomics Lab here at IFOM since a little bit more than 1 year. The idea is to study proteins, as says the name, meaning that we can analyze, identify, and quantify the proteome of a cell, or of a tissue, or of an organism. And this is clearly essential to understand the basic mechanism how a cell works, because the proteins are the key players of cellular life, let's say. And in particular, we are interested in mass spectrometry-based proteomics, which is a technology-based research, which is very essential to identify all the molecules or the proteins expressed in the cells at a given time. So we have, for instance, in the last year, we have studied the proteome profile of organelles, subcellular organelles of cells, or protein complexes, how they work, protein talking to each other, or protein-protein interaction, or how proteins are modified post-translationally. Meaning that, for instance, if we study phosphorylation, we can understand how the signaling pathway inside the cells are transmitted. And we have example of interesting function regulated by phosphorylation or summoylation or redox signaling, meaning how cysteine-containing proteins are modified. And each of these modifications can affect cellular function, behavior, or interaction with other proteins. Both things. So for sure we have our own research, but we also have very much interaction with other groups who are interested in using this technology. Because I think your question is pointing out the fact this is an essential step in understanding then how the basic mechanism of a cells are. So there are also many interaction with other groups in IFOM and also outside IFOM, of course, where we use our expertise to address specific questions. In several ways, I would say, because, so for instance, our project now is aimed at understanding how tumors tame the microenvironment to promote cellular growth and spreading. So what we are studying from the proteomic point of view, of course, is the secretome of a cancer cell. And in order to understand which molecules are related to a more aggressive phenotype. And this is very important in order to understand cancer. So there are, I would say, several applications to this. So one is clearly, if we found which proteins are dysregulated during cancer, this can be a target for a new drug, for instance. Or if we identify which proteins are the signature of aggressive phenotype of a different kind of cancer, this might also help in diagnose the proper tumor type, and so to identify new biomarkers that can be helpful for the diagnosis but also for the treatment. So there is nowadays a very relevant branch of medicine which is trying not to properly cure or address the proper therapy to the different tumor type, and this is based also on the proteome phenotype or profile of a specific patient. And I would say this is one of the most hot field in the moment, because of course the clinical application is very, very relevant. Yes, so protein profile could be the cancer profile, or also proteins which are, for instance, present in the blood, and that might help to address the cancer subtype or the proper therapy to this. So we have recently finished one work in lung cancer, and it is known that, for instance, there are tumors that respond better to certain kind of drugs or treatment. And by proteomic profile, we can discriminate which patients will respond to this drug and which will not respond. So also the medical doctor knows after reading the proteomic profile which drug is the best for this patient. Yes, well, it could be together or separate. So clearly this pretty much depends on the cancer type. So in this case, of course, there is also genetic predisposition, but there is also another way of reading it, which is the proteomic profile. So it can be in parallel or also separate. This depends how much we know of this tumor at the moment we are analyzing it. I think there are, well, two basic fields. One is more related to basic research, so clearly knowing the molecular players, and the other one is more on biomarker field. So relating to the molecular players, as you know, for instance, one limitation of proteomics now is in its, let's say, quite a large amount of samples, so we don't have a single cell resolution. And this, I think, is one of the way of the field where proteomics will develop, trying to really have the molecular signature of a single cells, so defining better all the different tumors. And the other way is more related to biomarker discovery or profiling, as I said before, and this is more, I would say, a technological problem, which means that we will need more user-friendly instrument and fast machine just to have them applied into clinics every day, which is not the case so far. But it will come, it will come. Yeah, now they start to be. So this was also a big problem in the past because it was not easy to translate the finding on, let's say, because proteomic studies are related to small population of patients, and then to translate them into real clinical practice, you need to validate in a larger cohort of patients with all the variability that I pointed out. Because one of the problems of the proteomics with respect to genomics is that the proteome is much more dynamic, and it changes a lot and more different from one person to the other one. But nowadays, this has come into clinical practice, so there are biomarkers, proteins that are used, and there are also ways to quickly validate them by, as I said, technological innovations.